different types of omics data. And you probably already kind of know what they are. There's genomic data, transcriptomic data. We'll soon get into metagenomic data. So there's all kinds of omic data. And it represents some biological phenomena. This biological phenomena drives processes that we call diseases. And they make all kinds of interactions between different cells, different tissues, and different even organisms that come from outside of our body. And a lot of the application of this type of data has to do with matching the right kind of data with the right kind of analysis and establishing the logic of how the two correspond to each other. So this program is called OmicsLogic because it focuses on the logical component of bioinformatics. Now, after you complete this course, which is an introduction to bioinformatics, you will be free to decide which direction do you want to take this into. One direction could be to apply bioinformatics to different types of biological problems, or another direction could be to maybe focus on the algorithmic component. Maybe you want to design better algorithms to find associations between different factors, or maybe you want to adapt different types of methods to novel technologies in the omics world to help process and make sense of these types of big data sets. So this program is like a gateway for you to then make further decisions about what you want to do with this information. But we also hope that in the context of this program, you will find how different types of informatics approaches of data storage, organization, analysis, visualization, and reporting can be applied to different types of biological examples. And so what we'll do today is we'll review what we have covered so far. And so far we've covered kind of an introduction to working with data, and then working with genomic data, and then now you are completing the working with transcriptomic data. But as you understand how these different types of data sets are similar or different, it's great to see the application of the different methods of analysis in the context of what we call projects. Before we jump into that, I want to remind you that throughout this course, it was myself, Beepsa, who's not mentioned here, but she's been playing an active role in helping kind of everybody walk through the content and find all the different resources that are available. Dr. Mohit Mazumdar, who has been primarily focused on updating Dr. Tuohy on how to manage the different types of content that we have. And Dr. Harpreet Kaur has been making sure that all of the assignments have been checked and you get responses to the different questions that you have. So we've all been trying to kind of work together to make sure that you've had so far an experience that is beneficial and useful to all of you. And I want to remind you that if you do have any further questions, you can reach out to us, and there are several different ways that you can do that. And please allow for some time for us to have time to get back to you in time. The way that this program was organized, just schedule-wise, is that we are right now right about here in the mid-assessment. And this mid-assessment is going to provide you with some kind of a self-evaluation for where you have gotten to in this process of this course. So we'll share after this session is over a survey, a form that you have to fill out that will help you kind of understand where you are. And just to kind of review what types of topics we have covered already, we looked at two languages that are very actively used in the domain of bioinformatics. This is R and Python. So again, this course is not going to make you necessarily proficient in either one, but it is going to give you an overview of how different methods could be packaged in different packages and libraries so that they could easily be called as functions, and how you can write scripts to import data, visualize data, and perform different types of analyses. Then we turn to genomic data, which is generated by next generation sequencing. And we looked at a few different examples of how this could be used for oncology studies and different types of pharmacogenomic examples and stuff like that. So then we looked at what's called transcriptomic data. And so transcriptomic data is different from genomic data because instead of DNA, it's generated from RNA. The RNA in humans comes from the process of transcription and translation ultimately, when the DNA is transcribed into mRNA. And the mRNA, messenger RNA, is what becomes eventually proteins, but it goes through several changes, including alternative splicing. And so when you look at the difference between next generation sequencing of DNA data and mRNA or RNA data, the analysis is also going to be different, but the same in the sense that you still have to map the short reads onto a reference. And that reference becomes the genome of whatever host you are sequencing. So in the case of the different types of applications of methods, there are going to be universal methods like mapping, alignment, normalization, etc. And there are going to be also specific methods, like a method for finding specific variants, like variant calling for genomic data, or methods for evaluating alternative splicing, which would be more relevant to mRNA because that goes through alternative splicing. Now, as you go through this course on Transcriptomics 1 and Transcriptomics 2, you will find that we use an example. And this example was the same paper that you started with the pre-assessment in, which was on patient-derived xenograft models. And this is a good project to use because it essentially shows how alignment or mapping of those reads to the reference genome can be used also as a way to study the interaction between the two tissues, the tumor and the microenvironment. And so we have this case study, which is one of the examples that we will use throughout this course to kind of understand the application of bioinformatics to a variety of different projects. So what we'll do today is we'll take a look at some additional projects, and we'll talk about how bioinformatics can be used to detect somatic mutations in cancer, to study drug resistance in tuberculosis, to understand viral adaptation and geolocalization of the Ebola virus, and look at other examples that have to do with both biomedical and non-biomedical topics. So we'll take a look at a few of these examples, but you also have access to a lot of different projects that we'll point you to towards the end of this session. So how do all of these program resources fit together? So what we hope to create in the OmicsLogic program is a way for someone who is completely new to bioinformatics to understand how different methods essentially help someone that has to deal with large quantities of data that ultimately have a biological interpretation using a number of different resources that you are likely to find in any kind of a bioinformatics lab. So these include some training materials that are all based on projects and research publications, because a large component of bioinformatics is being able to take data from somebody else, right? So when you are a bioinformatician, when you learn bioinformatics and then you apply it to a data set, more likely than not, that data is going to be owned by somebody else, which means that somebody else generated it in a specific way, has the metadata that you probably either don't have or need help understanding. And so we hope that through this process, you actually learn how to read research publications, and specifically the methods portion of the research publications, where the type of data is described, how it was prepared, what does it represent, and therefore you can plan your analysis. The second part is the platform. And it's a big data platform because many of the data sets that you'll end up using have thousands of samples across many conditions with hundreds of thousands of observations per individual sample. And because of that, in any lab, you will have access to some kind of a server, some kind of a centralized location that has all the necessary scripts and maybe some loaded data sets. And instead of having everything on your computer, you would have to separate the design of your analysis from the actual analysis process, which is conducted somewhere else. And so this is something that you have to get used to. You first understand what the data is and logically have to create a pipeline and then execute that pipeline and find all the necessary outputs to answer a specific question. So hopefully in examples like Transcriptomics 1, you were able to see how the pipeline produces maybe hundreds of individual files, and you have to know where to look to find the specific answer to a question that was posed to you. You also have to understand the logic behind the analysis so that you are running pipelines that make sense. The third resource is coding. So coding is a big part of bioinformatics because it helps you modify things from how they work for somebody else's project, essentially, to how they should work for your project. And so a lot of times people have designed these packages and libraries around specific applications. For example, maybe somebody designed a specific analysis package to study haplotype diversity of HIV viruses in a population. Now you're faced with a new challenge. It's COVID-19, and you have to apply the same logic, but to data that is a little bit different because, for example, HIV, evolutionary speaking, does not change as fast as COVID-19. And it also doesn't spread as fast as COVID-19. So while you have the similar approach, you have to tweak some parameters, modify the type of data that you're putting in, and also maybe change the way that you present your data visually so that it makes sense to your collaborators. So you're not speaking about HIV anymore, you're talking about COVID-19. So that's why it's important to understand that you are going to a lot of times work with scripts and packages and functions that somebody else has designed. So you have to read the code and understand what the code means. In some places, you will have to modify the code, for example, the type of input data, and then also tweak parameters along the way so that the outputs from your analysis are relevant and also meaningful. And then finally, we have this fourth component, which are projects. And so these projects really illustrate the application of all of these three different components to different types of biological or biomedical challenges that you're likely to find yourself applying these methods to. So first of all, on the platform, the types of analyses that are going to be useful for this program include analysis of gene expression, alignment of large whole genome sequencing data sets like you did with the variant calling pipeline in Genomics 1, analysis of phylogenetic structures for hundreds of samples of, for example, COVID-19 genomes. But also you have a lot of other tools that could be used in this omics kind of world, because ultimately the omics data oftentimes represents different sides of the same biological phenomena. So if you think about a problem like cancer, which is a systems biology problem, it's not just the tumor as a whole that is important for analysis. You actually have to look at individual cells that have specific variants and maybe have different types of properties affecting other cells around them. And so the genomic variants have an effect on transcription. Those probably have an effect on protein-protein interaction. That's where you want to focus for intervention, and maybe you are designing some kind of a molecule to stop that interaction. And so multiple types of omics and multiple types of approaches are going to typically be used in the context of the same project. And that's what the platform is built around. That's what the logic of the platform is. And so we hope that through different examples that you explored during this program, you are able to see both individual methods, but also how those methods fit together to help you study a biological phenomena from a systems perspective. The other resource that you probably saw was this application in R and Python. And so ultimately, popularity comes and goes, right? In bioinformatics, there's still a lot of methods that are written in Perl that, is a language that's been around for decades. And so as you look at the different types of popular or not as popular applications, I think what you will find is that they are all useful as long as they produce the types of results that you can interpret and you understand how they have been produced. And so the goal of these exercises that we are providing is to help you learn the language, help you see how this type of analysis is different from maybe other types of applications of coding, like maybe in web design or analysis of other types of data, because you have to deal with data that is maybe unique to the world of bioinformatics. For example, this kind of sequence data even though you can find sequences in text and natural language processing, they're very specific because they do have only 4 characters or maybe 22 amino acids or something like that. So they are very unique to the world of bioinformatics. Finally, the coursework that we've discussed so far in the course was an introduction to bioinformatics that we hope kind of provides you with an overview of different applications of bioinformatics, followed on by introductory courses in genomics and transcriptomics. And now we will also continue to explore what's called machine learning and how it applies to transcriptomic or other types of data, as well as metagenomic data, which is experiencing a tremendous growth of applications, because we see the applications of microbiome to anything between severity of COVID-19 infection to sensitivity to different foods, to acne and different types of skin diseases, and all kinds of other types of applications. So we hope that through this brief overview, you'll kind of get a little bit of a feel for what is different about this data and how potentially you could use this data in your different types of projects. So far, you have done, well, we hope that most of you have been able to look through Transcriptomics 1 and Transcriptomics 2 courses. So I'll just briefly comment on what these 2 courses include, and then I want to make sure that we pause and hear any questions that you have about this. The Transcriptomics courses are designed to kind of reiterate on some of the aspects of genomic data and genomic sequencing that you probably already read about in Introduction to Genomics and Genomics 1. So what's important about this is that in the traditional sense of RNA-Seq, you are trying to estimate an average of mRNA expression across a whole variety of conditions. And so several methods have been designed to make sure that the error in your input data is minimal, your variation from technical noise is addressed, and then you have some kind of signal that could quickly summarize information from thousands or even tens of thousands of points of information into something that you can easily grasp. And so these methods that we will discuss help you explore your data and then ask specific questions about the data. And by asking the question, what we mean is generating and testing a hypothesis. So to give you an overview of RNA-Seq, you probably learned this already, but we'll kind of go through the steps. The logic is that, first of all, your input data is pre-processed, or in other words, it is trimmed from different types of technical variation that you do not want to include in your analysis. The second step is mapping, when you map the data onto a reference genome. And here, potentially, there could be a number of different types of information that you're looking for. It could be only looking for known transcripts that have been reported and are put into some kind of a database or a file called the genome transfer format. Or you want to identify potentially novel types of alternative splicing events or transcripts that could inform some kind of deregulation that is typical for complex diseases like cancer. Ultimately, then you want to quantify the levels of expression so that you understand for each individual mRNA, how much of it do I detect in a particular sample or a group of samples. Now, based on the exploration of this table of expression, of the quantified levels of expression, you can now design a hypothesis. And that really has to do with how do you organize your data into meaningful groups? And then you have the methods of differential gene expression that you can use to study how this hypothesis applies to individual observations in your data. So let me ask you here the first question. When I talk about samples and observations, what do I mean when I say observations? What are they? And you can add your answer in the chat box. Let me just pull up the chat box here. Or you can unmute yourself if you want to answer. Okay, so Ken is saying sequence reads. Anyone else? Replication of a sample, says Trong. Okay, so when I say, again, I'll repeat the question. When I say samples, right, those are individual samples that I've taken, for example, biopsy samples, these are biological samples. But when I say observations within a sample, what does that mean? Does anyone else have any answers for this? Just maybe wait until we have a couple more. Okay, so Kendrick is saying trends with a question mark, trends. Anyone else? So one more answer and I'll move on. The data. Okay, so all fairly good questions. So let's start with Ken's answer. Sequence reads. So in fact, the evidence that I have is in the sequence reads, but those sequence reads came from some biological material. And so the biological material represents mRNA that was expressed, right? So the observations that I have are mRNA transcripts. And those transcripts typically come from gene sequences because in the transcript, we have exons and introns. Introns are spliced out, exons are spliced together, and we have gene sequences. So these are sequences of protein isoforms. So you can refer to these as transcripts or isoforms, and those are the observations. So when I do the next step, when I look at differential gene expression or I test my hypothesis, I'm actually testing that hypothesis each time for an individual observation or individual test on a gene or an mRNA transcript. Okay, Trong answered replication of a sample. So typically we do have replicates in our data set because for any biological process, there's individuality that we want to kind of get rid of. And so a lot of times we're looking at replicates of 3 or even 10 or more per condition. So we can get a better sense of what's common for that condition and not unique for that individual. But those are not the observations that within each sample, right? Those are observations that are different samples. Kendrick mentioned trends. I assume that this is trends in gene expression. So yes, those are kind of derivative values that we can analyze from RNA-seq data. And obviously this is the data, but just keep in mind that in any kind of an analysis, especially when you talk about hypothesis testing, you're going to be test your hypothesis on observations. And those observations are going to be genes in the case of RNA-seq data. So the final step is differential gene expression. And this is really where the traditional RNA-seq pipeline kind of ends. So traditionally, the pipeline is going to be consisting out of these 4 different steps. And if you master these 4 different steps, you can do a lot of interesting analysis with RNA-seq data. And hopefully we'll get to a few examples today to kind of illustrate that. So here's another question. When would you not consider using top hat? So first, maybe let me ask, which part of this is top hat? So pre-processing, mapping, quantification, or differential gene expression. Where in these 4 groups of methods does top hat land? Okay, Ken, correct, mapping. Okay, so, but when would you not consider using top hat? Thank you, Nahid. So here you have 4 different choices. 1, when you do not want to do exon detection, as with non-coding RNA. 2, when you have not run an error correction algorithm. 3, when mapping on a genome that lacks an annotation file. And 4, top hat is always the best choice. And you can use, yeah, okay. So Nahid answered number 3. when mapping on a genome that lacks an annotation file? So again, the question is, when would you not consider using top hat? So the answer from Nahid would be, when mapping on a genome that lacks an annotation file, you would not consider using top hat. Anyone else? So, I mean, this is not graded. So this is just, for us to have a discussion about what's right and what's wrong, make sure that you understand this correctly. So feel free to guess if you don't know. Okay, great. So everybody's guessing number one. So which is when you do not want to do exon detection. Okay, great. So that is correct. It's number one, because Top Hat is a collection of two different types of algorithms, bow tie and an exon detection and exon splicing detection. So a number of different steps that help us annotate novel transcripts based on detection of exon splicing, and then producing what's called a transcriptome. So whenever we do annotation of gene expression and we quantify gene expression, we can say, well, everybody's got the same gene expression. Everybody's genes are expressed in a similar way. So we have a database of expressed, 22,000 genes. We can just ask, is this one expressed? Yes or no. Or we can say, everybody's genome is expressed differently. So let us first prepare a transcriptome, which would be a new annotation for this particular genome that will tell us which genes and which combinations of exons are actually being expressed. And then we'll quantify all of our samples as a single data set so that we have transcriptomes compared across the samples that we have. So it is an in-depth approach. An alternative approach would be just mapping on transcripts, which would be bow tied to T, or you also have these non-mapping types of approaches like Salmon and Callisto that could be used without mapping onto the genome altogether. So why would I say that differential gene expression is kind of like your standard end to this logic of RNA-seq expression? Because typically, or historically, I probably should say, experiments were designed around simple comparisons. I have one set of samples that represents condition. I have another set of samples that represents control. And so when I want to understand a condition, I just compare it to the control. However, as people started generating more and more data, it started to be clear that you have a lot more variation. For example, if you have a condition like cancer, you can say, here's a healthy person and here's a person with cancer. But the healthy person is probably, maybe they're healthy from cancer, but they have diabetes. Maybe they're healthy from cancer or they have heart disease, right? So there's a range of things that we call healthy. And then there's also a range of severity for a condition like cancer. Maybe it's cancer stage 1, 2, 3, etc. Maybe it's different types of cancers, etc. So as a result, we need methods that deal with more variability than just grouping everything into these simple 1 or 2 type comparisons. And so one of the ways that we can do that is designing specific regression factors that can help us analyze relationships. And so what we are essentially saying is that we can study the effect of some factor that could be measured by observing only those genes where that effect is consistent with the level of a factor. So for example, if I have a factor of time and I have 3 time points, then I can see some trend that is only consistent with there being 3 time points and say, I only want to look for genes that have been affected by this kind of a factor. And so here we can analyze different types of more complex relationships. Specifically, not just to spot the obvious. For example, the obvious would be there are 3 factors or 3 levels of one factor called time, and the expression just goes up. That's a fairly simple type of a design that you can spot even without factor regression analysis. But sometimes you have interaction of factors. For example, I have somebody who is running and their temperature rises, and somebody who is drinking and the temperature goes down. So when you drink, the more you drink, the temperature goes down. The more you run, the temperature goes up. Now I combine the two, right? The person is running and drinking at the same time. What will I find, right? And so here, the question would be, well, how does the temperature vary across time? And maybe I have 100 people running, and I can say, well, I clearly see that when a person runs, the faster or the longer they run, the temperature rises. The more they drink, the temperature goes down. But when they do it together, maybe there's this unexpected effect. And so it's an effect of interaction. And that effect of interaction, which is one factor and the other having an influence together, could be unexpected. For example, temperature rises 2 times as fast as when a person is just running on its own. Or it takes 3 times as long for the temperature to rise to the same level because the person is drinking at the same time. And so those kinds of effects are really something that we can investigate using factor regression analysis. So to place all of these different methods into a logical kind of schematic, I would say that there are 4 different specific steps to this overall process. First of all, you deal with data that somebody has designed. So there was an experiment designed, potentially in a lab. Somebody said, I'm going to take these mice and do this to these mice and take these mice and do something else to them, and I will collect data on them. And so when you approach a data set, it's important to understand how that data set was prepared and what considerations to have about the experimental design. Many times when that data is collected, it is provided to you in this raw format. And this raw format, just like we saw with FASTQ files, is not anything that you can analyze. Essentially, it's just short sequences of reads. If you wanted to align them all to each other, you would fail because they come from completely different places of a genome. And so the first process is called data cleaning. And it's typically assumed that a bioinformatician or a data scientist will spend probably between 70 and 80% of their time just on cleaning and processing that data. So when you do data processing, it's important to understand what type of unwanted variation you want to eliminate. So what's noise? What kind of signal you want to preserve? So how do you want to represent the signal in your data? So let me ask you here a question. In RNA sequencing, you've learned about 3 methods of quantification of mRNA. Does anyone remember what they are? What are the 3 kind of metrics that are possible or typically used for pair-end read data for RNA-Seq? Okay, Ken is saying northern blotting. Trong is saying FPKM. Okay, so Ken, northern blotting is one of the methods to detect whether some presence or absence of mRNA, maybe quantify that, but that's not a quantification metric. So Trong is correct, FPKM is one of those. This is when I process my read data and map it onto the genome, I can then quantify the abundance of mRNA in specific metrics. And FPKM is one of them. Direct count or kind of raw read count is another one, correct? And so what's the third one? Does anyone else remember the third one? TPM, excellent. Okay, so FPKM, raw count, and TPM. So those are the 3 metrics. What is different between them? Raw count means that I just count how many reads I have. And so if you imagine one sample having a coverage of average across the genome of 1,000 and another one having 5, again, average across the whole genome, then this metric doesn't really make sense, right? Because here I just have a lot more reads. So why would I just count from and compare them between each other? So FPKM and TPM were designed to address this challenge. TPM simply is transcript per million. So it says, let's say you have 5 million reads and I have 10 million reads. So let's just divide it. You divide it by 5 and I divide it by 10. And so that way we will get a measurement of transcripts that is comparable, apples to apples. FPKM goes one step further and actually also accounts for fragment per kilobase per million, which means that it looks at the length of each individual gene. If you have a total of 5 million or 10 million, they're not distributed equally. If you have a longer gene, that longer gene, statistically speaking, is likely to have more reads fall on that region just by chance. And so I can also, since I'm counting transcripts or, in this case, genes, I can also incorporate this measurement of length. So that's about processing the data. There are several considerations, like we talked about top hat, right, is used to detect novel transcripts and splicing of exons. There's the levels of quantification. All of these are important measurements to prepare my data to accurately represent noise, the amount of signal that I have in a given sample, and produce it in a right kind of a metric for the next step, exploratory analysis. So can somebody give you, or give me, sorry, a method of exploratory analysis that you've learned in the transcriptomics courses so far? Just name any method that you've learned so far. And please, for all of those that are participating, again, there are no wrong answers here. This is just a review. So if your answer is wrong, that's okay. Okay, Ken is saying clustering. Anyone else? Does anyone else have any exploratory analysis methods in mind? Anyone else? Okay, well, you probably remember that you can explore data using statistical characterization of data properties, right? So what's a property of data that we could compare between samples? For example, distribution, or we can take something like maximum or minimum value, right? Or we can look at skewness, or we can look at median or mean, right? So these are just different characteristics of each individual data point. If we have a sample, we have some data set for that sample, and we can say, okay, well, is the distribution of this sample different from the distribution of another sample? Or if I take a group of samples, is the distribution between the two different? What if the mean is different, right? What if I have 3 samples and the mean is low, and another 3 and the mean is high? What does that tell me about the different samples? So the answer to that is that in a controlled experiment, either that is a known factor in advance. So somebody said, I'm going to take, cells that are living in this rich environment with a lot of food, and I'm kind of expecting all of their activity to be upregulated. Okay, and Ken is saying they may be from different populations. Yes, they are probably from two different conditions. And if I see that, I need to ask the question, why is that? And so either that could be by design. So for example, two different populations that I want to compare, they have different means, that's expected, I want to compare that. Or it could be noise. And sometimes in controlled experiments, you would just by chance see that one is more contaminated than the other, or one can be a little bit more noisy than the other. And so then you want to normalize that data. So exploratory data analysis is not just knowing something about your data, it's asking active questions about your data to address those questions before you turn them over to what's called the actual analysis step. So clustering is a method. Method is to characterize the data from the standpoint of statistical properties. And that could be done using visualization or summary statistics. And also you can use methods like principal component analysis that reduce dimensionality so that we can actually make sense of unwanted variation like outliers or maybe groups that are too much overlapping and things like that. So once we've prepared our data and it's ready for analysis, we go into data analysis, and we already talked about statistical models like differential gene expression uses a statistical model, or factor regression analysis uses a statistical model. Or we can also turn to more advanced methods that we will learn in Transcriptomics 3, like classification and predictive analysis and things like that. And so ultimately, the end of this process is to inference, right? What do we actually infer from the data? How do we interpret the results to talk about what biological processes are important for this condition, or how does this biological condition compare to another condition? So that part is very important there as well. But at least for the process of processing, exploratory analysis, and actual comparative data analysis, I think this is what we have covered up until now. How does this apply to different types of problems in biomedical research? So biomedical research, and we've been kind of doing a lot with biomedical research so far, could be divided up into three categories: basic research and translational research. And translational research could be in clinical or an industry application. So when we talk about basic research, imagine yourself working in somebody's lab. They specialize in some specific topic, and they typically want to understand the mechanism behind some specific type of observation. So they want to understand, how does this actually work? How does it happen? And typically what they want to understand are specific principles, biological associations, methods of regulation. So they're asking questions about the basic biology behind what is being observed. And a lot of the data sets that we have been dealing with, they are kind of in that spectrum of basic research. Another type of an application, clinical application, means that you can actually take what you have found from the basic level and apply it to ask clinical questions. And clinical questions typically have to do with either diagnostics, how do I diagnose a disease, or therapeutics, how do I prescribe treatment? And so around those two topics, there's a lot of translational research that helps doctors be more precise in how they diagnose and more personalized in how they prescribe treatments. And then there's the pharmaceutical and the biotech industry. Typically, what the industry is interested in is some kind of a topic that could be industrialized, right? And so, for example, if we have biomarkers and we can screen millions of people and know that for 3 biomarkers that we measure, this is the drug that's going to be 24 or 36% efficient every time, this is a win, right? This is a strategic advantage. So I can implement the strategy and kind of industrialize the process. A lot of times that has to do with the discovery, right? So how can I find a drug that solves a problem in a better way? how can I apply that drug to a certain group of people for patient stratification and for biomarker-driven targeted therapy. And I also can think about how to address other types of questions like stimulating the system for like the immune system, for example, in our body and prepare it for some kind of an adverse effect. So those are some applications in biomedical research. So now I'm going to ask a question. It's not related to anything technical. This is a personal question, so feel free to answer it, but I hope you're not, again, this is not for grading, so feel free to answer this just for discussion. What types of projects that you've read about so far in this course, from Introduction to Bioinformatics, to then Genomics, to then Transcriptomics, what types of projects or what types of applications from these three categories are you most interested in? Basic research, that would be number 1. 2 would be clinical, or 3, pharmaceutical and or biotechnology. So maybe I can see if everyone can answer this question. So we just kind of get a survey for what kind of interests do you personally have? Okay, thank you, Arnold. Okay, so Truong, we actually didn't have a 4. There's only 3: research, clinical, or pharma and biotech. Okay, Kevin, pharma. Kendrick and Nahid, clinical. Truong, biotech. Okay. Jaretta, research and clinical. Ken, clinical, probably leveraging research studies. Okay. Okay, is that everyone? Let's see. We have Arnold, Connor, Truong, Sean, Kevin, Nahid, Kendrick, Jaretta, Ken. Okay, I think that's everyone. All right, great. So let's talk about some of the projects that you could potentially think about as you study these different methods. Again, remember that this whole program is designed in such a way that we will first learn about different types of omics data, then we will learn about different types of processing and analysis of data, and then eventually we hope that by the end of this program, you will be able to formulate a bioinformatics project proposal. So in other words, you will propose kind of your own study. If you were free to do whatever you want to do for the next 6 months or a year, what would you actually spend the time working on? And it doesn't mean that you actually have to work on it. This is just to say that you know how to do it. You can translate your idea to a data set and a method of analysis. So let's talk about that really quickly. So the types of bioinformatics research projects that we're going to be talking about are called research driven by data. So these are going to be data-driven research projects. And Bipsa, if you're there, could you please post the link to making the most out of your research projects in bioinformatics? It's just a reference. Okay, thank you. So when you think about a project, you have to consider several different things. And we already know what you're interested in. So this makes that part of it easy. But that's the first part. So these are topics that you know and care about. And that means that, can you prepare a few slides explaining the topic background, or can you see yourself spending a few months researching this topic, right? So you have to have some sort of motivation to move in this direction. The second part of it is what will be interesting to your audience. So when you consider selecting a topic, you want to think about, are there enough people that will actually care to listen about this topic? You can test this easily by trying to tell your parents, your siblings, or your friends about the topic that you're interested about and hearing what they have to say. If no one is interested to listen, no one understands what you're talking about, maybe that's too niche, it's too narrow. Maybe if this is only interesting to the 2 friends that always are interested in what you're interested in, that means that it's not a generally kind of interesting project. And the other part of it is, do you hear the questions or discussion about this topic? So for example, is it covered in the media? Like for example, the vaccines for COVID-19, which is a both clinical and a biotechnology topic, have been discussed at the level of genomic data for so long in the media that anybody could care and listen to that topic. But if you take a topic like reproduction of chlamydia in sub-Saharan Africa, maybe not as many people would be interested in that. So just think about those topics as you select your project idea. And then an important critical factor that a lot of times people forget about is that there is a sufficient body of data available on this topic. So this means both publications that describe this so that you can base your analysis on some kind of a reference, and is there a particular data set, or typically a number of different data sets that you can start exploring when you consider selecting that topic. And so in the intersection of those things that you know and care about, what will be interesting to your audience, and sufficiently studied and data available, you will find the best and most impactful topic that you want to focus on. And so as you move into the second phase of this program, think about what types of data sets, what types of project ideas and designs and analysis methods that you feel are interesting and potentially aligned with your research interests could be useful for you to focus on. At the end of the program, you will complete a research study proposal on how you can apply some of the things that you've learned to a data set of your own, or to one of the projects that we include in this program. So then what's important is to make sure that you know the data, you know why the data was generated, and you know kind of what to do with this data. And this typically falls into these three categories of data sources. So there could be primary research, secondary research, and tertiary research. And typically primary research is the research that you produce raw or original data on. So this is your first-hand observation. And this is typically where you will source your data. So that means that somebody had a problem or a question, and they designed their hypothesis around that, or they designed their experiment to generate data around that problem or question. And then you will move into secondary kind of research, which is an analysis and interpretation of somebody else's data. So that's what we hopefully will want to do, is we would want to review and critique and maybe improve upon somebody else's data that they've generated. And maybe that means adding new analysis and maybe making novel interpretation. And then tertiary, which is kind of the next step, typically, we're not going to go into that, is when you want to say, okay, well, everybody's interested in this particular type of cancer. Let me collect a whole bunch of samples from this domain and make it into an easily searchable and curated database. And so that might include data, tools, and collections. So that's not what we will do, but we will try to find primary data sets and review some secondary materials. Potentially, we will use some tertiary research by looking into compilations and databases to source new data. But this is important to understand because ultimately you want to go through sources that describe the context of your project and provide you with a general background. And that means that if you think of a topic, just think of what are the primary data sets, what are review articles that are available on this topic, and what are some databases that I could search to reference whatever I'm going to do with this data. Now, from that general background, you want to move to a more specific and focused hypothesis. And the way you do this is you first want to review available sources and categorize them. For example, if we talk about different categories, right? We talked about mechanistic basic research, we talked about clinical and biotech, we talked about different types of applications and translation of this research. You can categorize them like that. Is this an article about the basic mechanism of transcription regulation for this, 2 genes? Or is this an attempt to develop a biomarker assay that helps me diagnose people better, right? So you can categorize them into different categories. Then you want to identify gaps in unanswered questions, maybe controversy, a debate, maybe some kind of gap in existing research. So some people are saying, we have done this analysis and it kind of points in this direction, but there's obviously a gap. More needs to be investigated. Or somebody is saying, I found these 3 biomarkers, but they only work 50% of the time, right? So what about the other 50%? So you can find gaps in existing body of research. And that will lead you to define your question or hypothesis. So when you define your question or hypothesis, it will combine two things. The first is, what is that general background? And two, what is your specific hypothesis? And that's a good place to start writing your abstract. So let's take a look at an abstract of a paper and see, what does this actually tell us about why the people selected their project, what they intended to do, and what was their conclusion from this research article. So this is a publication that is used in Transcriptomics 3. So if you already started Transcriptomics 3, as I saw some of you did, you'll be more familiar with this data set. If you have not yet started, this is the link to this article. Let me share this link with you. And you will find this link referenced in Transcriptomics 3 as well. Okay, so this article is called Modeling Precision Treatment of Breast Cancer. And as you can see, the abstract is separated here into three sections: the background, the results, and the conclusions. So the background is that first-generation molecular profiles for human breast cancers have enabled the identification of features that can predict therapeutic response. However, little is known about how the various data types can best be combined to yield optimal predictors. So if you go back to the previous slide, what is the gap or the controversy or that specific focus that they found in existing research that they wanted to address in this publication? Okay. Does anyone have an answer? Just the first sentence here. What is the background that they found a gap in the existing body of research and wanted to focus on that particular gap to answer that question? Does anyone have? Okay, so Sean is saying optimal predictors. Okay, so how good the predictors are, maybe that's a gap, right? How various data types can be best combined to yield optimal predictors. Okay, so I think Kevin is identifying this correctly. They are saying that we already know that individual features can predict therapeutic response, but little is known about, right? So that's the gap. Little is known about how the various data types can be best combined to yield optimal predictors. So it is optimal predictors, but it's in the context of data integration. So what kind of data did they use? Primary, secondary, or tertiary? What kind of research is this? Did they generate their own data? Did they take somebody else's data? How did they kind of start this study? What kind of data did they use? All right, excellent. So they did not generate their own data. This is not their own data. They used somebody else's data. In fact, they say we used collections of breast cancer. They used some people not only generated the data, they also organized and collected them into a searchable database potentially, right? So they've used somebody else's data that was already organized for them. So collections of breast cancer cell lines mirror many aspects of breast cancer molecular pathobiology, and measurements of their omic and biological therapeutic responses are well suited for development of strategies to identify the most predictive molecular feature sets. Let me ask you here, what do they mean by most predictive molecular feature sets? What could that mean? What are we trying to predict here? Okay, so Sean is saying antigens for therapeutic response. Anyone else? Which gene expressions can predict cancer? Cell line of breast, Nahid is saying. Okay, so anyone else? Okay, the correct answer is that they are trying to predict drug response. So you can put drugs on cell lines, see how they respond, and ask the question, what if I don't put any drug on a cell line? Can I just predict what the response would be, knowing the molecular profiles, knowing gene expression, knowing variants? Could I just look at a patient, take their molecular features, and predict what kind of a response they will have to a particular drug, right? So this is an application of an understanding of a basic mechanism of, let's say, mRNA transcription regulation to the ability of assigning a therapeutic to a particular patient. Now, it does have that diagnostic component as well, because I could also identify different types of breast cancer from doing this analysis. But this is the project. We recommend that you start reading this publication and kind of going through the Transcriptomics 3 course to really learn about how they use the transcriptomic data and what kinds of methods, as you can see described right here, to do this kind of predictive analysis. And we want to make sure that you are aware of other types of projects that have been designed to kind of explain some of the analysis methods and the data sets. And a lot of these are going to be focused on cancer. So you will see analysis of changing immune responses in cancer, modeling cancer precision medicine, etc. And these projects are going to help you understand an article, the methods that is used in the article, and what types of outcomes you might expect from the analysis. So all of these projects are essentially done to help you read through publications that have been digested by our team, extracted into types of data and types of methods, so that potentially you can develop novel and independent projects that are similar to these. So as you look through this project and go through the Transcriptomics 3 course and the rest of the material that we'll do in this course, kind of use them as examples that you can enhance and apply your understanding of this, this analysis to whatever problem you're interested in. So I'm gonna stop right here. I think we do have Mohit and Bipsa here that can give you a brief overview of your progress and kind of where everybody is, and kind of explain this form that we will share with you for you to kind of give us some, evaluate your own progress. But before we do that, do you have any questions about what we covered in this session? And in fact, any questions about the material so far? Just the content itself, or the different methods that you maybe didn't understand, or maybe some question that you didn't understand that needs to be addressed. So we didn't receive any questions ahead of this session, so we didn't really know if you didn't have any questions, but if you do, we can answer those now. So Mohit or Bipsa, could you guys take us through the progress?
Yes, sure, Ilya. Give me a moment. I'll be sharing my screen. Just one moment. I'm sorry. I'll be sharing in a minute. Okay. So far, I've seen that most of you have been very active during this session. So let us get started with the progress review here. So this is a collective review of progress for everybody as part of the session. So we can see here what are the different accounts that you have used. By accounts here, I mean the different platforms. So the pink here is representing the OmicsLogic platform. The gray represents the Bioinfo server. And the yellow is represented by the Edu platform. So this is the cumulative progress report for everyone in the group. Now let us take a look at some of the individual progress. So before we move on to the individual progress, I see that Kendrick has been in the rank 2 on the OmicsLogic platform. So very good work, Kendrick, on the OmicsLogic platform. And he has 11,465 points. Now, in the upcoming individual review, we are not doing what you're going to be seeing, that the points from the OmicsLogic is not added. So you are only going to be seeing the edu points and the server points out there. So some of the points may not match up, but we will be sending you the weekly updates soon. So first we have Kendrick, of course. So here we can see Kendrick's profile, the number of points that he has, as well as this is the progress report from the edu platform that tracks the progress across different time points where you might have been active. So this just shows the activity that you have been doing across these days and not actually the courses that you have taken. So well done, Kendrick. And as I move through this, if you have any questions, please let me know.
Excuse me, Beef, so I have a question.
Yes, please.
Is it your plan to go through every student's status here? It may not be appropriate for to share everyone's status with everybody in the class.
All right, so okay, so we will not go through everybody's progress then. So this is all that we had to share. So yeah. Then I will stop sharing. Yeah. Okay. So
I think if any member of the course wishes to inquire about their present status, we'll be happy to share that with them.
Sure. All right. Okay. So I will not share this then because we had prepared an in-depth review of each person, but we'll not do that for today. So if you all have any questions regarding your points or any other related things, please let me know. And I think Truong here had a question. So was it answered that the course does not help us master R code and Python?
Yeah, I can answer that. So the training in R and Python includes many aspects of basic kind of, terminology and the different types of data and the different types of, writing your own functions. And then, there's a whole lot that goes into, I would say, proficiency in R and Python. What this training will provide you with is kind of like an overview of some of the important methods that you would need if you want to use them to analyze omics data, and specifically kind of accompanying some of the tutorials that we've had with the different project examples. So if you are interested in becoming more kind of proficient with these, I think there are some Code Academy or Data Camp and other types of tutorials and collections of tutorials, and potentially you might want to consider spending 3 to 6 months just working on one of these languages to become more proficient and kind of professionally ready for various coding challenges that you might face. But with what you will get in this course, I think you'll get a taste of the different languages, different methods and functions that are applicable to omics data, and the types of analyses that we do for the projects that we've selected for this course.
Thank you, Ilya. And James, if you would have something to add further.
Yeah, I just wanted to say I apologize for interrupting your.
That is absolutely all right. That is absolutely all right.
But just because of FERPA regulations, we can't share all that data. But I want to encourage members of the class here, if you have any question about your status or where your course standing, to please contact Beepsa personally. And also I want to say, we're getting towards halfway in the course right now. And I want to encourage you to come forward to raise any questions you have. Please don't be shy. There are no medals for struggling in silence. So, just make sure that you voice any concerns or challenges you have either to the faculty at Pine Biotech or to me. And of course, I'll just pass them on to Pine Biotech anyway. So please don't be shy in coming forward. So that's all I have. Back to you, Beepsa.
Yeah.
Well, I'll just maybe end this with the reflection.
Sure,
for the
Ilya,
intro.
please go ahead. And I also wanted to add that, the forum is also open for any questions. So if you are not comfortable just asking us, you can also post it on the forum. So I will also share the forum link again. Ilya, please go ahead. Thank you so much.
Okay. This is the reflection. It's not really that long, but you'll see here, we do ask for your valid email address. In fact, just try to use the same email address as you use on the other platforms here. And then here we have just how much time you spend for the different courses and the different activities that we've done. And then here, what of the following best describe your understanding of concepts presented in the course? So just kind of like, how do you feel about things? Are you confused? Do you feel confident that you understand everything? Which of the following best describes how prepared you feel for the concepts and skills presented in the course? Which of the following best describes your interest in conducting future research in bioinformatics? And which of the following describes your interest in pursuing additional training in bioinformatics? So similar to what was already asked about R and Python, is there a particular skill that you feel you want to now become more proficient in? And maybe there's a particular topic like cancer biology, let's say you want to study, or maybe there's something else that you've gotten out of this. Here, we ask you to kind of self-evaluate your skills, the way you feel about reading and analyzing, different data and, doing different methods of analysis that we've done here. And I've covered today a couple of project examples, but there are more that we have, including some on TCGA, which is the Cancer Genome Atlas, COVID-19, Ebola virus, etc. Or are you thinking about more of something of your own that we haven't covered here? So this is just to get a sense of how you feel about going about a project proposal. And sometimes people prefer to work together. So if you do have a teammate in mind already, you want to work with somebody else and develop a proposal together with somebody else, that's perfectly fine. This is something that you will have plenty of time to work on and think about. So this doesn't mean that you have to kind of submit a project proposal already. And this is a bonus thing. So if you do submit a project proposal, it will be evaluated, but as a bonus to the overall training. And this is something that you can answer right here. And any other comments, right? So here it says other comments that may be helpful for assigning research project teams. If you feel like you don't want to work with anybody else, or you just want to focus on one aspect, we ask that at minimum, you try one of the projects that we provide. The next kind of level is you want to take one of the projects, but also add on to it by maybe doing your own literature review. And the next stage is actually if you want to formulate a research strategy or how you will find and analyze data, to propose that plan. So those are some of the options that you have. So this is the form. We'll also email it to you separately and post it on the forum. So that's all I had. Thank you.
Thank you so much, Ilya. So thank you all for attending today's session. And I do hope that we have been able to answer any questions that you may have. So please continue to ask any doubts and get them cleared. And I look forward to seeing you in the upcoming session. So we will try to schedule it at a time which is more appropriate for everyone to be able to join. I do hope I saw that everybody has joined in today's session. But if there is any problem in time, so we will try to make sure that we can arrange for a session then. And this session, as it is recorded, will also send everybody the recording of this session. So thank you again, and thank you, James.
Thank you. Thank you.
Thank you. And thank you, Ilya and Dr. Mohit, and everybody else for attending today's session. So we will be ending today. Thank you.
Great. Thanks a lot. Bye-bye now.